Good day, everyone! I am Katrina Kayabyab, and the topic that we will be discussing to you for today is the monohybrid cross. At the end of the lesson, students are expected to explain the terms dominant, recessive, homozygous, heterozygous, phenotype, and genotype. Predict the results of simple crosses with expected ratios of 3:1 and 1:1, using the terms homozygous and heterozygous. Use genetic diagrams to solve problems using monohybrid inheritance. Our discussion will be divided into 4 parts. The contents that I will be going over include the founder of this study, the terms we need to be familiarized with, what exactly is monohybrid cross, and of course, demonstrating how to solve problems using the monohybrid inheritance. Make sure to listen to the end of the discussion because we will be having an interactive practice in the end of the video. Before we start learning about our topic, let us first familiarize ourselves with the founder. Gregor Mendel was a monk who lived during the mid-late 18th. He is the first person to lay the mathematical foundation of the science of genetics in what we now call as Mendelism. Because of this, he is considered as our father of genetics. He was interested in discovering how traits are inherited in the offspring of sexually reproducing organisms. Therefore, he devised on the process we call as monohybrid cross to answer the question of how does a single trait be passed from parent to offspring. Okay, so now we know our founder for monohybrid cross. Let's familiarize ourselves with the terms that we need to be aware of to learn in this study. A gene is a portion of DNA that contains the genetic code for the trait. Now, a trait is a characteristic that can be passed down from one generation to another. For each trait, we get some of the information from both the parents. Because of this, we may inherit different forms of the same trait. These different forms are called alleles. Alleles are one of 2 or more alternative forms of a gene that arise by mutation and are found at the same place on a chromosome. A phenotype is the express trait. It is the outward appearance or visible character of an organism. Basically, it refers to the expression of the trait. Meanwhile, the genotype is the genetic makeup of an organism. Basically, it refers to the exact pairing of alleles. For example, dwarf pea plants have genotype lowercase tt, while a tall plant may have genotype either uppercase tt or 1 uppercase T and lowercase t. Every organism has at least 2 alleles for every gene or trait. These 2 alleles have a dominant to recessive relationship. Think of it like superheroes and their sidekicks. The hero or the dominant is stronger, and the sidekick or the recessive is weaker. But they are both relevant. The dominant is always shows if present and is represented with capital letters, while the recessive only shows if both present and is represented with lowercase letters. Coming from the word homo, meaning the same, homozygous means having the same 2 alleles at a given locus on homologous chromosomes. Let us apply these 2 alleles as an example. When we see 2 uppercase letters, such as TT, we would refer to that as homozygous dominant. When we have lowercase tt, it is referred to homozygous recessive. Likewise, heterozygous, coming from the word hetero, meaning different or distinct. This means having 2 different alleles at a given locus on a pair of homologous chromosomes. For example, we can take one uppercase T and one lowercase t. We can refer to this as heterozygous because this. You don't need to assign it to recessive or dominant designation because it's the only heterozygous option. Therefore, it has no further distinction. Now that we are familiarized with the different terms for the topic, let us get on to the definition of monohybrid cross. The monohybrid cross is a mating event between two parents that are heterozygous in respect to the single gene. This means that each parent carries two different alleles for the gene. Mono, coming from the word one, refers to only a single gene, and hybrid, meaning mix, refers that each parent has two different alleles.
Thank you, Katrina. To further understand, let's use a classic Mendelian example using pea plants. The pea plants in this example have two possible colors, yellow and green. The yellow phenotype comes from the presence of the dominant allele, and the green phenotype is from the recessive allele. If the genotypes are homozygous dominant or heterozygous, the peas will appear color yellow, and if the plant is homozygous recessive, the peas will appear green. So, if you have two heterozygous parents and you want to predict the genotypes and phenotypes their offspring will have, you will have to set up a Punnett square. A Punnett square is a tool used to predict the probable outcomes of a genetic cross. To make a Punnett square, we draw a square and draw a cross inside of it. To fill up the Punnett square, we must first separate each other's parents' alleles. On the top, we segregate and record the female alleles, and in the left, we segregate and record the male alleles. Now, to fill it up, we will put the parents' alleles in all possible combinations. We put the dad's allele here, and the mom alleles goes here, which forms a homozygous dominant genotype. Mom's alleles go here, and dad's alleles goes here, forming a heterozygous genotype. And in this box, dad donates his allele here, and mom alleles go down here, creating another heterozygous genotype. And in this last square, mom and dad both donate a recessive allele, forming a homozygous recessive. Now that we know how to make a Punnett square, let's apply it to our Mendelian example. 1/4 of the offspring will become homozygous dominant, 2/4 or 1/2 of the offspring will be heterozygous, and 1/4 of the offspring will become homozygous recessive. The genotype ratio is 1:2:1 ratio, and their genotype percentage is 25%, 75%, and 25%. Always take note that each square is 25%. And for the phenotype, you can see that 3/4 of the offspring will have yellow peas and 1/4 of the offspring will have green peas. This phenotype ratio is 3:1, while our phenotype percentage is 75% and 25%.